Excellent. Thank you, and thank you to the organizers for having me speak today. I should present myself. My name is Silas, and I'm a postdoc in Alex Dunton's lab and in Jeff Axelrod's lab at Stanford. Imagine these are cells. These Pepsi cans are cells, and you can sort them upside down, and we like this, and we define that as apical basal polarity. You can also sort them to a second degree, left-right, and we call that proximal distal or planar cell polarity. We can look at an epithelial sheet, here 8 cells, and if we take these 2 cells down here and look into these and see how they build polarity, we can imagine those 2 cells here on the side. Initially, we have 2 groups of proteins equally distributed across the membranes. We have a green group termed north, and we have a purple group termed south. On one side, the green group promotes itself and inhibits the other group. On the other side, in the other cell, the opposite behavior occurs. Across the membrane, there's also interaction. The same group inhibits itself, but promotes the other group. This results in 2 groups, in 2 polarities being built up inside the cells, a south pole and a north pole, or in this case, we call this proximal and distal. Inside the proximal group, we find the membrane protein Wang and the cytosolic protein Pregel. And inside the distal group, we find the membrane protein Frizzled and the 2 cytosolic proteins Disheveled and Diego. Across the membrane, we find a flamingo acting as a bridge connecting the cells. This is the planar cell polarity signaling pathway, and it's responsible for building up this second direction, proximal distal, perpendicular to apical basal. Now, imagine you view the sheet of cells from top, and you look down on wings, on fly wings. In fly wings, we see hairs all pointing distally, similar to the Pepsi cans I showed in the beginning. Also here, I'm showing in the cartoon on the left a hair coming out at the distal side, pointing distal. However, if we mutate any of the PCP proteins, we see swirls in the hair pattern, as shown here on the right. This not only occurs in flies. These proteins are conserved across mammals, and we see similar phenomena in, for example, the mouse ear. Here we have the inner mouse ear cells. In the wild type, they're oriented up like the Pepsi cans, but in the PCP mutants, they are all messed up. And they hear loss hearing. So why do animals care about planar polarity? I mentioned just 2 concrete examples here, but why is this at all important? Like all our organs have an inside out, and the cells need to know what's the direction towards the tip of the fingers compared to the shoulders, and also the direction of the internal flow in our organs, or of whether it's liquid or air. So having mutations like these can result in severe developmental diseases and defects such as neural tube defects, heart defects, swapped organ directions, and it has implications for wound healing and cancer. Knowing the direction is important for the cells. Here I zoom in and imagine the fly sheet again. Here you see what is this, 8 cells in the fly sheet. So this is something we can do in flies. We can label Wang in YFP here on the right in a subset of the cells, and in a neighboring subset of the cells, we can label the same proteins in cyan. This allows us, the reason why this is cool is because it allows us to scan down the membrane here shown with the arrows and look at the intensity coming from one cell to that particular membrane, and also looking at what is the fraction that comes from the other cell, since now we are separating the green and the purple. So we can do a line scan that will look something like this, and we see, for this particular line scan, we see 3 clusters. PCP form clusters along the cell membrane. Here they accumulate into clusters, and we find the highest degree of asymmetry inside these clusters. Here you see an accumulation of Wang on the proximal side and less varying on the distal side. So we ask, why do clusters matter for planar polarity? How do cells build a polarity, planar polarity? And this is kind of the ultimate goal of my postdoc. I'm not going to tell you the full answer today, but what I can tell you is what is the size distribution of PCP clusters. And here we kind of imagine 3 scenarios. Here a cartoon showing the interaction between 2 cells. One possibility could be that these clusters that you see here on the right tend to all come in one fixed size. An alternative could be that there's a coarsening mechanism going on at all times, that clusters, they merge, and eventually they merge into one big cluster. However, at any intermediate size, you will find a bimodal distribution with a lack of intermediate size clusters. This is coarsening. A third option could be that clusters, they not only fuse, they also split apart, there's fission going on. And in that case, if that's the case for PCP, we will find a power law distribution of clusters. But which one of these is the case? We didn't know when we started the experiment. So to answer this, I study the fly ring under the TURF scope, here shown varying GFP in live wings. You see the wing sac. It's inside a sac. You see also wing cells with expressing varying GFP. You can identify the wing cells. And you may even notice, like this cell is the tip of the wing. And you may even see how varying is polarized into these lines. You can see folds in the wing sac. You can see folds in the wing. We can now zoom in on any particular group of cells and get a high-resolution image of the clusters. And already here we see a huge number of clusters in these around the cells, varying clusters, PCP clusters. What I do is that I bleach this in time, and I thereby get a bleaching trajectory like this one of the cluster pointed out here with a red arrow. And by doing that, I can identify a single blinking fluorophore late in the bleaching trace, and thereby get a size measure of a fluorophore at this particular location in the live wing. I can, that comes up there. I can go back, and we can even, in this trace, identify where we have 2 fluorophores on and 3 fluorophores on. Thereby, we can find the best estimate of the step size of the fluorophore. And we can now back calculate how many fluorophores or how many molecules are in this particular cluster at the beginning. And for this one, it will turn out to be 8. We can do that for all the clusters in the wing. Like what I show up here. Just take any of the clusters without double counting, and we can get a distribution of cluster sizes out. Here I did it for Diego, one of the PCP proteins highlighted in the cartoon down on the right. And what we notice here is that PCP the cluster size follows an exponential distribution, with most clusters being small and larger clusters are less and less occurring. I should explain that the lowercase n is we should get the lowercase n is the number of biological replicates, and uppercase n is the number of clusters I looked at, and then mu is the mean average cluster size in the fit, in the exponential fit. Okay, so here we got an answer to the question, and this is probably scientists in a nutshell. We try to think of all the possibilities we can come up with, and then it's none of them. So this is an exponential distribution. So this indicates a single underlying growth function, meaning that the energy required to add a new monomer is independent of the cluster size. We can continue and ask, what is the stoichiometry of PCP clusters? And the cartoon I showed earlier on, that's kind of the textbook view of PCP, with double number of flamingos compared to the other proteins. However, we've never looked with high-resolution images like the ones I'm doing here. And we can do that, just continue what I showed you before, do it for the other proteins, and we find equal the number of disheveled, frizzled, and prickled. However, as expected, there's more flamingo compared to the other ones, roughly factor 2, so that makes sense. However, varying is surprising. There's significantly more varying compared to the other proteins. And that means we will have to change the textbook view down there to having not only one varying, but possibly 4 varyings on the proximal side. So flight genetics allow me to make clonal and boundary experiments like the ones I'm showing here, with a subset of the cells having expressed GFP, and another subset on the top not having GFP. And thereby, I can identify the size of proximal clusters and the size of distal clusters, separate the two sides. And I can analyze along these lines, and I can thereby ask how many flamingo are there on the two sides. And I do find equal amount of flamingo on the two sides as expected. And I also find that prickle is it's almost 100% polarized, with the most being on the proximal side. And frizzle is on the other side, so this is all good. However, for varying, the most is on the proximal side, but there are equal the amount of varying distally as there are frizzle distally. So we have to change the cartoon below again from moving one varying from the proximal side to the distal side. So this makes us change the cartoon. I explained this. I think, so now we can continue and ask other questions. How does it develop with aging? How do these clusters come up if we look at them early in time? This here is right before pre-hair formation, but we can look even earlier and we see reduced varying. So what I'm going to conclude is that cluster size follows an exponential distribution for PCP clusters, indicating a single underlying function. Varying plays a very more nuanced role than people have thought earlier, with 2 populations of varying, one on the distant side and a larger population on the proximal side. And what I now want to do is that I want to continue this question here. Why do clusters matter for planar polarity? And we have some mutations in the DIX domain, in the Sheffield, that we have been working with for some while now, where we hope to perturb the cluster, make slight perturbations to the cluster size without disrupting cluster formation at all. This is a strong collaboration between Alex Starn's lab and the Jeff Axelrod lab. Thank you.